Hi, I'd like to thank you guys for taking the time to come in and listen to my oral presentation. Today I'm going to be giving an overview on an article that is specifically looking at genome testing. And the name of the article is The Association Between Type 2 Diabetes Loci and the Measurements of Fatness. Real quick, just a little bit about the journal in itself. The journal I pulled the article from is called PLOS One, and this particular journal has an impact factor of 3.73 in the first year, and it has an impact factor of 4.244 in a 5-year time frame. So it makes it a substantial journal to be able to get published into. So this actually is something we're going to look at. So the significance of this particular article was quite interesting. When we look at type 2 diabetes, or we define type 2 diabetes, it's basically a disturbance in our metabolic system. And so it is a disruption in our ability to be able to metabolize carbohydrates, fats, proteins, and produce insulin levels to help significantly reduce the glucose levels. Unfortunately, what we're starting to see, though, is there's quite a bit of an expansion in individuals that are being diagnosed with type 2 diabetes. On a global basis, we have roughly about 170 million individuals that are currently type 2 diabetic. But unfortunately, we're looking at an explosive number to almost 350 or a little more million by 2030. So we're starting to see a rise in this particular chronic disease. And it has an effect on our social health aspects. And so we know that currently, as research goes, that there are a couple of genes or loci that have been identified to be associated with type 2 diabetes when they run genomic testings. So the purpose of this particular article is to flip the thought process. Do these genes, or do these individuals who carry these type 2 diabetic loci, manipulations of the genes, are they more susceptible to increasing fat content on the body? We already know that increased fat or obesity is related to type 2 diabetes. Now we want to see, is type 2 diabetes precursor related to the increase in the body fat index? And so that's the purpose of the study. Part of what we see is that the journal does recognize that, or the article does recognize that 2 of the genes associated with type 2 diabetes is also involved with the development of fat tissue. So the hypothesis is that these individuals that are diagnosed with these gene manipulations will also have an increased rate of fat mass index and body mass index. We also know that the FTO gene is in relation and directly involved in regulation of the food intake. And this is also a gene that is associated with type 2 diabetes, which is another reason why the hypothesis is that these genes associated with type 2 diabetes will also be associated with fat mass index. So the researchers decided to utilize roughly 3,000 participants. This population is an interesting population. It is currently located in the southwest region of the Netherlands. They are considered genetically isolated because the villages were established in the 1800s, and there was very little migration in or out of the particular region. So the population started at 400 individuals and is now exploded to over 2,000. So this particular population has a very close genetic signature. So they utilized these individuals for this study. And what they did was they identified the polymorphins that were associated with type 2 diabetes. And they measured these particular genes in the participants. And at that point, they also identified the 2 additional genes that we already know are established with type 2 diabetes pathways. So these were the ones that they looked at in regards to relations for our fat mass. There's roughly about 9 of them. So what were they trying to do? Well, they took these genes and they wanted to see if there was an association with a number of different areas. The first one was the fat mass index. They also wanted to know, do these genes have an association with increased fat percentages? And they were using a DEXA to actually do the measurements on those particular items. They wanted to see if these genes had an association with an increased body mass index. They also wanted to see if there would be a difference in the waist circumference and the waist and hip ratio if these individuals had these genes. And so what they did is they determined that the power for the study on a nominal level would be 0.05 on the p-value, which meant that there would be a 97% significance rate, that there would be an association between the genes and these particular areas of interest that the researchers were looking at. So what did they find? Well, if you remember, the FTO gene was in regulation, or was involved with food intake and the regulation of food intake. They found that this gene was directly associated with BMI. They also found a significant association with fat mass index, and they also found a significant association with waist circumference. Now, on the body fat percentages, there was a borderline significance when it came to this particular gene. So individuals that had this gene, the FTO gene, they saw a significant increase in these particular physiological responses. However, on the other ones, there really wasn't much of a significant show of change for the physiological aspects. So the thought was, perhaps there's a combination that we should be looking at. Maybe there's a combination of genes that we should be considering to see if there would be a significant influence on changes of those physiological respects. And after they looked at it, they really found that there were no significant changes for any of the combinations associated with the other genes of the for type 2 diabetes. Really, the only significant gene that had any bearing on the physiological effects of the body happened to be this FTO gene. So, as you can see, they've had quite a bit of substantial significance behind it. Do you guys have any questions on the results of the study? No? Well, I'd like to thank you all for your time today. Have a great day.